Evening, everyone. We always got to wait a few minutes for the crowd to roll in, or not a few minutes, but a few moments here. It's going to be a good one tonight. Biome makers, this is going to be good. It's right up my alley. I want to know what's going on out there in the field. So let's let people are coming in. That's good. We got some good numbers coming in. Awesome. I apologize for last week, everyone. I just could not pull it off. I've been traveling too much, and I just couldn't do it. So I apologize for not being on last week. Um, just one thing I want to say. We're starting to get requests for the not only the audio version that you can easily do on podcasts, but people want to see the video version as well. So Rachel's been doing her research, and we're going to start pushing some. I mean, eventually all of them, but she's going to start pushing some up to the cloud as both audio and video. So one day we'll be able to then just see, because I like to see, when I hear in the back is great, but I like to see the body language and what are people talking about and their focus and all that. So that's just me. But anyway, all right, let's go. Giddy up. Here we go. Let's go. We got a great one tonight. Biomakers. I'm going to start. I'm going to start with Mary. My usual question, Mary. What is on your mind right now?
That's a good question. So I am driving right now to back from Nebraska to Colorado, where I am living. And I just met with a big group of agronomists out here, and I'm listening to a book on my way home called The Lion Tracker's Guide to Life. And it's an interesting title, but just driving back through the plains and listening to this book that really is about finding your track in life and reconnecting with nature and listening to the signs that nature gives you. I think that there's a lot of synergy within agriculture and like our work at Biomakers and what we were talking about today with agronomists. And so it's like this interesting synergy, like it's this lion tracker, right? Who's just like really dropped in with the animals and following animals and finding lions in the safari. But there's all of these synergies around really dropping in and paying attention to the ecosystem and what's going on.
Yeah.
And that's what I'm thinking a lot about right now. And I think it's really relevant to what we're doing, what we do at Biomakers and our work within agriculture. So
Yeah.
that's what's on my mind, just like the intersections of tuning back in with the natural world and how that is where we're headed in agriculture and the direction to keep growing food.
Isn't that weird how there's so many parallels in life? So that's awesome. So thank you, Mary. We'll be back to you in a minute. Gus, well, I'm going to ask you the same question. You've had a little bit more time to think about it. What's on your mind right now, Gus?
Well, unfortunately, I don't think it's going to be quite as eloquent or as great of a connection to regenerative ag as Marie, but there is a connection there. On what's been on my mind is my next batch of microbrew beer and kombucha it's something that I've been experimenting with and kind of a little bit of a side hobby that does have some relevance to agriculture, given that some of the same some of the same microbial species, bacteria, fungi, yeasts that we look at and measure every day at Biomakers are some of the same important players in the fermentation process of beer, wine, kombucha, all sorts of fermented drinks and fruits. And the other thing on my mind is just is gift giving season was coming with the holidays approaching. That's always something that as Thanksgiving gets here, I'm like, it's time to get those holiday lists filled out and get the loved ones some gifts. So those are really two things top of my mind, but with the topic Marie brought up, a lot more to think about and unravel in our conversation today, probably.
Yeah, that's awesome. Thank you. And Marie, I apologize. I pronounced your name wrong at the beginning. I'm sorry.
It's okay. You wouldn't be the first one in my life or today, and for that fact.
I apologize. So it'll be Marie the rest of the evening. So Marie, give us your background. I mean, how You're not very old. I'm not going to ask you how old you are, but I know you're not very old. What made what clicked? When did you say, okay, I'm going to do what I'm trying to do today? When did this happen?
Well, it happened in 2016. I'm an engineer by training and had worked in the renewable energy space and oil and gas space, and
Oh
like always
wow.
really had this desire to work in nature some way or capacity. And we grew up camping and hiking and like looking at things under a microscope. My dad was a scientist, science teacher. So, but it was in 2016 that I was kind of like, I really don't want to climb stack towers anymore and measure emissions from the top of refineries. Uh, I want to work in something that is meaningful, that connects the work that I want to do out, like just being outside and um, working with ecosystems and the connection to people and humans. And um, I was talking to a friend who was like a climbing buddy at the time in Colorado, Bobby Gill, who, if you follow Savory Institute, know him. And I was like, Bobby, I just don't know what I'm doing. Like,
Yeah.
I am working as an environmental consultant, which I thought was like my dream job, but like I'm doing air compliance numbers on a spreadsheet. And he was like, do you know what regenerative agriculture is? And just kind of like cracked me open to agriculture, really introduced me to like agriculture in general. And he um, just kind of talked to me about the millions and millions of acres that are managed under agricultural production. And it really clicked for me in that moment that like working in agriculture is at the intersection of science, nature, spirit, like the spiritual connection around like working with the land,
Yeah.
um, and people and communities, and like how every time we sit down with our family and eat, we're connecting to nature and we're connecting to agricultural systems and we're connecting to the people in rural America that are growing our food, and we're connecting to the 900 million acres that raise animals and plants across the United States.
Right.
Uh, and so that was like the really big like connection for me, was like this is everything that I'm interested in. And working outside, uh working with growers is like just the most salt of the earth amazing people on the planet.
Yeah.
And so it was like in that moment, within the next year, I was farming and ranching myself. I started working on a goat ranch and was just like all in. I was like, I'm going to engineer full time until I figure out how to transition into working in agriculture full time. And I moved onto a farm and just kind of like dove headfirst into figuring out how to wrangle goats. And
Well, that's great. So farm girl and were you a farm girl then?
not growing up. No,
Not
I
growing
just,
up, no.
it
But
was
you
when I
are
was introduced to
now.
like agriculture. I was like, oh, I started talking to farmers and I was like, I have no idea what I'm talking about.
But
And then I was like,
but
I
that
need
can
to
be
do
a
this
good thing
myself.
sometimes. That can be a good thing because then you don't have any of the bad habits of all the other types of farming that tend to destroy things. So,
Yes, yes, and you also don't have like the context of how hard it is to
yeah,
do all of the things that we're talking about, right?
no,
Um, so I think there's like both worlds, and I think that like that's what I get really excited about bridging, is like it's really the context and the human element of changing things and implementing new practices and how hard that is. And when I started working on this goat ranch, they were using like antibiotic grain, and
mm,
animals weren't rotated, and like everybody was just in this tight knit group, and they had a lot of deaths every time they had babies. They had a lot of weird defects, and like there was some funky stuff going on. And as soon as we transitioned, it was hard to like transition away from that into a fully grass fed system, rotational grazing, yada yada. It took
yeah,
many years to start seeing the changes and like get the pastures back into a system where we could even do that without hauling in a lot of hay. But the health in the animals was like noticeable in year one. So anyways, I could go down a tangent, but like um so yes, it's like great to have this fresh perspective not growing up on a farm, and also having farmed now myself for a few years, also like the realness
Yeah.
of the hard work that it is.
Yeah. Understanding. Yeah. Yeah.
So
Uh, Claudia's on. Claudia's on every night. Thank you, Claudia. Happy Thanksgiving to you. Also, how's your back? I think you had some back surgery, if I recall. So hopefully everything's coming along well with that. Gus, all right, same question. When did you know?
Well, I first of all would say I followed a pretty similar path as Marie. I think we have a lot of parallels in our experiences and our path towards regenerative ag. I would say it was actually also around the same time, 2016, 2017, that I kind of was becoming more aware of how much I wanted to dive into regenerative ag. I found out I started to really get into it during the first years of my higher education, and I actually was wanting to go into studying land management and natural resource management. I always kind of was interested in looking at the bigger picture as far as forestry and ecosystem management, and not just thinking about them as in terms of their value, but also the ecosystem value they provide, and some of the unique aspects that go into forestry and silviculture. But as I started to work research jobs in forestry and vegetative management, it was very difficult to separate out some of the human-induced impacts. And most of my research experience was in Florida. I went to University of Florida for my undergrad. But agriculture was always a big kind of an underlying factor in what we were trying to do as far as land management restoration. Growing up in Florida, a lot of issues as far as water quality and runoff and algae blooms, and also a challenging environment with sandy soils. And I started working for a horticultural sciences lab that kind of got me in the realm of agriculture. And as I learned about the idea of applying some of the concepts we learn in forestry and ecosystem management to agriculture, it really clicked with me. And I think that a big part of it was also starting to get involved in the agricultural community. In Gainesville, Florida, there's a lot of small farms and a lot of community supported agriculture, CSAs, and farmers markets, and a lot of folks trying to do things regeneratively, but on a smaller scale. And that was when I really went around the second year of my undergrad, started to gravitate towards the idea of regenerative systems and cover crops and no-till and such. And also I did have my family, actually, my parents grew up in Wisconsin. My dad grew up doing farm labor, and my grandparents were both farmers. So I had a little bit of that background. But what I got out of it growing up, my dad, he hated the farm labor he did growing up and wanted to get out of it. So pursued a job outside the field of agriculture. And he likes to joke, well, I worked hard so that you could get out of agriculture, and then you went ahead and hopped back in. But I kind of come from not so much a hard ag background, more of an ecological or ecosystem focused lens. And it's been really exciting. And just I went on to get my master's at University of Texas in an agroecology program. That was a unique opportunity because it was one of the few programs I could even find that offered it didn't just focus on hard agronomy, but focused on systems thinking and some of both the human and the agricultural and the environmental dimensions of agriculture. And then the other thing that really pulled me in was the human health dimension, both human health and human sociology, which learning about some of the facts around declines in nutritional quality, and also some of the incredible improvements I've even seen myself working in research on organic cropping systems, some of the higher bricks levels and quality that can be achieved when the soil is healthy and crops aren't just being reliant on chemical fertilizers and crop protection products. So that's kind of the direction that pulled me full circle from environmental management more into regenerative ag.
Yeah. Oh, that's awesome. I love those stories. Thank you. Well, let's, I want to hear perspectives from both, but let's talk about biome makers now. That's what we're all here to I want to hear what you've got going on, and I don't care who starts. Tell us what your role is at Biome Makers, and what's Biome Makers doing for the regenerative movement.
Rick, before we jump into Biomakers, can we hear a little bit from you, like what's on your mind tonight, and how you got to where you are?
You want to flip the table? Okay, all right, we can do that, Marie. All right,
a little bit before we jump into biomakers, because that's
Yeah. Okay. I want to tell you, you know, the thing that really got me aware of this was erosion. So I started looking at regenerative practices. Of course, I didn't know what they were called then, but we started looking at those practices as a defensive mechanism to stop or slow down erosion, whether it be wind or water, okay? And then as we get into this and we realize the power that these cover crops have if you let them do what they were intended to do. And what I mean by that is if you've got a cereal grain and you want it to grow and create biomass and then have the ability to roll down for mechanical termination, you've got to let it grow to maturity. You can't come in and spray it when it's this tall with Roundup on the first warm day of spring. So once we realized that, then I realized, Marie, that cover crops were an offensive juggernaut and that we can now start to pull away these inputs. And that's exactly what we did. I mean, we took a systematic approach to it. You know, I started with 200 acres and I took the recommendation of the agronomist and I said, okay, great. I'm now going to take that recommendation on this 200 acres. We're going to cut it in half. And then we're going to see what happens. Well, that then showed that we could either maintain yield, if not increase yield, with 50% of the inputs. Then it's like, whoa, wait a minute. Is this really possible? And then you do it again, and then you bring in another 200 acres, you do it again, and then you're like, we're going all the way. And we're not reckless. I mean, we do Haney testing. I still love Rick's test. I think it's one of the best tests out there. And we then decided that there's way more important things here than chasing yield. So we need to chase return on our investment, and we need to chase human health. And so then my wife and I were talking about it. Carol wanted me to go organic way longer than I did. I just couldn't figure out a way to do it that wouldn't jeopardize the farm. But once you sit down, Marie, and you start to look, at least for us, and this is all about context, but you look at our family and you think, an uncle died of cancer, my wife had cancer, my nephew had cancer, an aunt had cancer, there's diabetes. I mean, and then it's just like, whoa, wait a minute here, what's going on? So then you say to yourself, I don't want our kids to be around this stuff, and I don't want our grandchildren to be around this stuff. So we're going to now go regenerative organic and just see how far we can take it. So that was kind of a quickie journey there, Marie, but that's the gist of it.
It's awesome. It's nice to hear context before we dive into like what Biomakers does, because we can tie it all back. And so thanks for sharing.
You're welcome. So you just start us off. What's Marie's capacity and what's Biomakers up to?
So at Biomakers, I am an account manager or business development manager. We wear a lot of hats in young companies like this.
Sure.
But what I do primarily is help our growers, agronomists, retail partners, ag input companies understand where our technology is best fit. Because like you mentioned before we, dove into this conversation, our technology at Biomakers, we do a lot of different things, but the basis of our technology is extracting DNA from the soil biology, creating functional reports that can help growers, agronomists, retailers, input manufacturers understand what's going on in the soil in order to inform decisions. And so you mentioned the Haney test, right? Any fertility management decisions are made on farm by a fertility analysis. And so what we're saying is biological decisions on farm should be made with a biological analysis. And
Exactly.
so, yep. And so that's what, in a very small nutshell, what Biomakers is doing is providing the biological analysis and a functional assessment, something that's really easy to understand and digest for the industry to make decisions with around biology.
Yeah, I'm going to tell you. Well, I'm going to tell you my story about biomakers after Gus goes. So, um, remind me, Marie, because my mind, I'm already thinking about four more questions. So remind me to come back to this, okay?
Sure.
Gus, tell us what your capacity is at biomakers, and how do you see biomakers moving, moving further into this regenerative space?
Sure. So I'm a technical agronomist with Biomakers. Like Marie said, we have pretty loose titles at a tech startup, but typically that's what I go by, an agronomist. So my role is mainly focused on interpreting our soil biology tests, helping educate our clients on how to leverage and use these tests for agronomic insights and management decisions, and also helping just with field technical questions on what sort of agronomic questions we can and can't answer, how we can go about designing sampling projects to answer them, setting up trials and such. So that's mainly my role, is really as a classical agronomist. But given my background more on the biological and agroecology side, I'm mainly focused on interpreting our soil biology tests alongside some of the Haney tests and others that we offer within our lab network. And as far as the second part of your question, biomakers moving forward, that I could probably spend the next 45 minutes just talking about that
Yeah.
alone. But
Oh,
I
yeah.
think Marie would agree that really one of the most exciting parts of our work and really moving forward. I mean, regenerative ag, even in my short career, we've seen so much momentum take off in the last 5 to 10 years. And what's really exciting is just being able to, we're really just starting to scratch the surface of what we can do with some of this data. And a lot of I always think back to kind of the route that chemical fertility followed. A lot of the chemical fertility tests still commonly used in the US today have been around, 50, 40 plus years. And
Right.
we have a lot of data backing them up. We have these calibration curves and, these yield calibrations that have been done to hone in on a regional basis, the levels of PK, micronutrients, and in some cases nitrogen that are recommended. But with biologicals, with biostimulants, biologicals, and cover crops and such, soil health focused management practices, we don't really have that breadth of data backing up how we make decisions. That we really haven't had the technology to measure it until tests like the Haney test came along, and now genomics in the last 5-10 years is finally getting cheap enough and scalable enough to apply the same kind of way that we apply 23andMe and looking at the human genome. We can now look at that microbial life by extracting the DNA in the soil. And really the exciting thing is taking that analytics and kind of like we've developed yield curves for chemical fertility measurements, we now also have AI and machine learning, which is part of our process for making these functional measurements in the soil that Marie mentioned, being able to quantify microbes, but also essentially sort them into functional groups of based on what they do relevant to crop production. And what we're really excited that we're already kind of getting started on this. I can't go into too much detail, but some of the preliminary projects we're working on and have on the horizon involve prescribing and using our data to prescribe biological solutions based on the microbial tests. So and then what's really exciting is before joining Biomakers, I worked in the biological product space. And well, the big thing you run into is the snake oil moniker. Biological might work great 2 or 3 times, but then it doesn't get a yield bump with a grower or doesn't show a response, and suddenly they've lost faith. But the problem is, just like you wouldn't use the same fertilizer or even the same fertilizer form, type, or application rate everywhere, you wouldn't even use the same lime everywhere, depending on certain factors and your soil chemical fertility tests. With biologicals and cover crops, it's the same way, the same solution, the same biostimulants, cover crop mix. It's just not going to work as a silver bullet everywhere. So getting the data behind those decisions and being able to use the largest soil biology database in the world and leverage that to prescribe and make some of these decisions with the data behind it, that's really the exciting direction that we're going at Biomakers.
Yeah, and see, I've been talking about this for a couple of years, and you're going to be able to figure out, okay, this farm in central Illinois has got a water hemp problem, okay? And you're going to figure out what biological presence needs to either be there or disrupt something that's there that creates the environment for that water hemp to want to germinate in. because it's only going to germinate if it knows it can grow and survive and do everything. Has only one purpose in this planet. One purpose is to procreate. That's what everything's purpose is. And if that environment is not in that right position, that water hemp seed won't do anything this year. It'll wait till next year. So once you guys figure out those triggers, this is going to be game over, I think. And then you combine that with a cover crop package of certain species of exudates. I think we now can manage a system with absolutely zero inputs and be totally successful. What do you guys think?
Yeah, I mean, I I agree on a few things. What I'll add is that so there's like, it's really exciting about where we're headed, but what we can do today is also really cool. And so that's kind of what I was thinking about touching on, is that, so this water hemp thing that you're talking about, right? We could
Yeah.
talk about water hemp and weeds. We could talk about disease pressure. We could talk about nutrient uptake. We could talk about all of these issues on farm
Sure.
that really impact ROI, right? That you were talking
Sure.
about, that impact ROI on farm. And so the future where we're headed is like, yeah, wanting to like get really nitty gritty on what exactly, what product might be the best use. But what we can do today is really cool in identifying, when you compare, let's say you have one field that is ridden with water hemp every year, and you have another field that water hemp never grows. The seeds there, right? Like the fields are
Yeah.
right next to each other. And I
Yeah.
saw this in our pasture all the time. There'd be one patch where there would be like toad flax like crazy, and then another patch that would just look beautiful. And so what we can do today is take samples in both of those locations and look at the difference and identify some of the key root causes for
Right.
differences in disease, differences in yield, differences in weed pressure, and help growers identify some of those root cause issues when you compare fields side by side. And then inform, that's what can inform either the grower or the agronomist to say, okay, so the difference between these two fields is magnesium mobilization and abiotic stress tolerance. Well, what can we do then to address those root causes to mitigate that issue? And weeds
Yeah.
is a really great example. If any of your followers follow Nicole Masters' work, she talks about reading your weeds, and that every weed does have a purpose and what is it doing there. And so we can drill down into what is the purpose of that weed there, and how can we remove its purpose,
Right.
right?
Yeah.
Without a product, or without like a necessarily an input, but like how can we understand why it's there and mitigate the issue?
Yeah.
So
See, I think this is fascinating because you're right. I've been talking about this for 2 or 3 years, but it's here now. It's here today. You guys
mhm,
are doing it. So I want to tell you my little quick what I came up with one day. Um, I came up with a couple of things. We're going to talk about one of them in a little bit, but this is different than what you guys haven't heard this. Um, I wanted to know I wanted to know what was the inherent biology in certain locations on our farm. So I went to areas on the farm that had not been touched or damaged by humans yet. So a wood somewhere that's always been a woods, a waterway, or a piece of a corner of a field that is a grass and has always been a grass for as long as my dad can remember. We went to those spots and pulled samples and took an essay, a biological essay of those samples, and then went out to those neighboring fields and took another biological essay, compared the two, and then said, okay, what are we missing here between the field and the inherent? And then there's people out there that can grow the biology that we need, and we filled those holes in. So now we have fields that have inherent biology in them based on what your study told us from those inherent samples. To me, that is huge.
yeah, that's pretty cool. It gets down to like working with nature, right? Like there's so much. I hear people talk a lot about, there's a lot of hunters in agriculture. You
Hmm.
go and you're out in the field and you're hunting, and like all of these things are growing, and it's like, what did you have to fertilize any of that stuff?
No.
um, and yeah, so and so the biology is really doing a lot for us that we just haven't had a way of measuring, and we have a culture of like needing the data and needing the science, and so that's what I get excited about. That's what we can provide now, is like we can show you what is there and what's not there, and then you can,
Yeah.
see with the data.
So, okay, I've always wondered, I know I'm assuming there is a collection point somewhere that's the main brain of this thing. And if someone identifies a species, then I assume it's being registered somewhere, and then you can pull from this database and update your algorithm that's looking for this biology. I'm assuming that's happening, right?
That's a pretty accurate portrayal of it. Yeah, we're compiling a database, a taxonomic database of the names of those species and their gene sequences, specifically the gene sequences that we refer to as biomarkers, which allow us to identify them, well agreed upon sections of DNA that can tell us code, essentially barcode or code for the species. What's really cool on that topic, Rick, with some of our metrics, so like you just described, we're compiling these lists of microbes that we pick up in samples. And what our data science team has done is, because they have this giant expanse of samples, they can even we've looked at which microbes seem to get along well because they have high levels of co-occurrence. They seem to synergize, cooperate, form essentially little communities, symbiotic communities. Just microbes don't just exist in isolation. They just, same way they interact with the crop root, they interact with each other.
Humans.
Yeah, same as humans, and kind of in some ways it's similar to like a food web. The connections with predator and prey, different trophic levels. But what's cool is, with this compilation of this database, we can see which microbes get along well, and then which ones don't get along well, which ones show levels of competition, co-exclusion, where they either fit the same niche and they don't get along because of that, or maybe they're natural predator and prey. So what we've done is, we do have one metric that's a soil quality score that you may recall from your samples, and that's based on partially that factor, the symbiosis, or how well these microbes interact ecologically. And we've seen in regenerative soils, our data science team published a paper looking at vineyards that were managed regeneratively or with biodynamic practices, as they refer to it in Europe, because this was an international study. Then organic, or sort of transitional farms that may use some combination of more regenerative and more conventional practices. And then they sampled a lot of conventional, intensively managed farms. And of course, as you would expect, we saw that the cooperation and synergy increased as you had more regenerative practices integrated into the programs for those vineyards. So that's one of the beautiful aspects of compiling this database, is the data as a whole is much stronger than individual, than the individual parts that make up that database. And that's part of what goes into being able to predict and use AI to drive predictions, is looking not only at the levels of microbes, but also their functions and how they interact.
Yeah. This is awesome, folks. If you've got a question, I mean, now's the time. We got two great folks, young folks here, work for Biomakers. I mean, this is the future right here. What we're talking about, it's here now, and it's only going to get better. All right. So you guys want to talk about the study the idea that I came up with? And okay, let's go there. Let me set it all up. I was talking one day to my good friend John Kempf, and I love John. He's really smart, and I just love the way he looks at things. And John and I were talking about foxtail, because foxtail is an issue that we have on our farm right now. I think it's a lot to do with progression, but there's other things at play here too, so we'll go there. That's fine. And John's concern was, well, Rick, I wonder, since you're no-till and you've not applied any synthetic fertilizers, any inputs of any kind for the most part, I wonder if maybe your soils are becoming stratified. And I'm like, well, that could be because maybe we've got a concentration of calcium, at the wrong spot in the profile, and that's helping contribute to the foxtail. So I then came up with the idea to let's go out I had another good friend of mine, Mitchell Hora, and his company, Continuum Ag. They were coming through the neighborhood, so I had them come. They pull soil samples for me every year, and we do the Haney test. And I said, guys, I want to do something different here. I want to pull I want to pull a core on every inch increment. So however many cores you have to pull of just the first inch, I think they took 20 of the probes, and they took just the 1 inch out of each top of the probe. It took 20 of them to get a sample. Then they took 20 to 2 inch, 20 to 3, 20 to 4, and so on and so on. And we did an inch increment from 1 through 6. And then they took soil samples of just a standard soil sample, 1 to 6, and then 6 to 12. And then I wanted a Haney test ran, and I wanted a Biomakers test ran. And now that gets us up to where we are. So, Gus, do you have, you've got control of the screen if you want to share some slides. I think I've set this up good enough. So now what we're going to see is, and by the way, Marie, I need to know what B crop means. So now's a good time to explain that. Would you tell us?
Yeah, so the company's Biomakers, right? And we can talk about that name too, but B crop in specific, the idea behind B crop and the feeling behind it is that we, we want to understand what it's like to be the crop in the soil.
Oh,
So it's like,
cool.
yeah, like on a biological level, like a major part of soil biology is the interaction that it has with the root exudates and the relationship that it has with the plant. And that's like the synergy that we need to understand. So that's where B crop came from. So we're B crop samples, B crop tests. We're understanding what it is to be like the crop in the soil.
And that's talking to us. I get it. I love it. That's great.
Yeah.
All right. All right, Gus. Here, let's see what Mark. Hey, Mark, how you doing this evening? How does biodynamic organic compare to? Oh, wait a minute, I lost it. Where's that? Where's my chat go? Okay, here we go. How does biodynamic organic compare to no-till regen organic system? Which is higher in nutrient density output or other differences? You got any experience with that? Or I mean, you got enough information to give an answer on that?
Yeah, I can take a shot. And if anybody else has more context, you can. So Mark, I don't know where you're from, but in the United States, biodynamic is actually a certification, and it involves some celestial, okay, in Wisconsin, some celestial practices around like planting with the calendar. It also, but I think at a high level, I think the practices are probably similar, like regenerative organic and biodynamic. They're two different certification bodies. One has been, biodynamic has been around for a long time. I think like, so that, I mean, I think they're probably pretty similar in terms of like practices in modalities. They just might have different routes or methodologies behind them potentially,
Yeah.
but I think the practices are probably pretty
But
similar.
Marie, can you guys do nutrient density testing? I mean, if someone sent you some a quart bag of some corn seed, could you do a nutrient density test?
We don't at Biomakers, no, because we're
Okay.
just looking at biology, not
Okay.
not the like chemical makeup or constituents.
Hey, Mark, that's a great question. And we'll have to find the answer to that because that's kind of intriguing, really. Um, and
I
I think I know where you're going with that, but yeah, curious about the tiller. Yeah, that's where I figured you were going on this.
Yeah.
Uh, and I would be interested to see that. But I'll tell you, Mark, let's hang on here just a minute and let's let these guys dive into my results. And I think this might kind of answer your question here. Go ahead, Gus. Great
Are you
question, though.
Are you able to see the slides
Yeah.
up on your screen? Okay, great. Great. So this is just really quick overview, and we have a few slides that I at the end, just in case we have more time to elaborate on the details. But there's a lot of depth in our samples, so we're going to kind of stick to the take-home message, which was focused on compaction. So here was that sampling design Rick mentioned. So each of these was a composite sample taken at the 1-inch increments, and then one sample composite samples taken across 0 to 6 and 6 to 12. With the real main focus of seeing if there's a compaction curve or any changes. One of the interesting things with compaction is there's microbial functions that we look at that are directly related to it. And I have this slide here. I know it's a little wordy, and this is probably sort of a refresher or already very familiar concept to I think a lot of your audience, Rick. So I'll try to be quick. Yeah. But with compaction, when we think about it, the big concern that can influence biology is oxygen and air permeability. With compacted soil, we're going to have a lower portion that can be dedicated to that air pore space. And when significant compaction occurs, this can cause the populations of microbes that rely on what we call aerobic respiration, essentially the same sort of breathing we do, oxygen-dependent breathing, taking in oxygen, breathing out CO2. And those microbes can decline due to compaction when there's not a lot of oxygen available for them to breathe. Some microbes may also switch to other metabolic processes if they can, when there's not enough oxygen. So some of those other metabolic processes, which we look at aerobic respiration on our B-crop test, but we also look at fermentation and methanogenesis. And those are some of those alternative metabolic processes that may increase when there's less oxygen available. These metabolic processes not requiring oxygen we tend to see jumps in these functions when there's compaction. And we've seen similar concerns play out, Rick, where folks send in samples. Usually it's not every inch. I think this is maybe the first time we've seen that, but usually it's every 3 inches or every 6 inches, maybe down to 18 or so. And sometimes we do see vast differences, the aerobic respiration declining at depth, and fermentation and methanogenesis off the charts due to those anaerobic zones. So the other important thing to keep in mind is this last bullet point, that generally speaking, and this is very general, but what a lot of soil health experts and especially compost experts will tell you is that aerobic microbes are generally more favorable to soil health processes than anaerobic microbes, which can include some of your pathogens. But I don't like to make generalizations because I know a lot of those anaerobic microbes are beneficial as well. It's just from a general standpoint, the reason that compost producers focus on aerating their compost effectively and ensuring a good balance of moisture and air is so that they can harbor those aerobic populations, which help with nutrient cycling and other beneficial functions we look at. So just with these factors around compaction in mind, this is why we looked at some of those carbon-focused metabolic processes that involve the flux of carbon in the soil. And this also does relate to building soil carbon. But what we see here on the right is this is one of our analytical tools, which is available on our website for any client. And this is what we refer to as our heat map tool. It's not necessarily a geographical map, but more of just a choropleth style display ranking the levels of microbes in each of these functional groups represented by the columns. So each column represents a different carbon-related microbial function. So we're taking the microbes detected in a sample and essentially sorting them by the functions they perform relevant to carbon. Carbon fixation. I wasn't really surprised to see that in the medium to high range in Rick's samples, because those are a lot of your beneficial microbes that can help directly sequester carbon. And in no-till cover crop fields, we expect to see pretty good numbers. And one thing I should mention. The schematic rating these samples from very low, represented by the bright yellow, to very high in the dark blue, that represents how these microbial functional groups compare to our database of soil samples. So that's kind of like a standard soil fertility test. We've developed a database and a standard rating system for these functional groups. And generally speaking, with certain functions, we want them to be higher, and others, generally lower. But with nutrient cycling, generally most of them we prefer higher. When we look at aerobic respiration here, again, this is the one that tends in healthy soils with optimal oxygen permeability and solid soil aggregation and structure. Aerobic respiration tends to be pretty good, like we see here. And that's and this, I should also make a connection to the Haney test here. The same way the Haney test, the Haney test measures respiration, but in a different way. They measure it, of course, with a CO2 burst test, looking at the amount of CO2 emitted by those aerobic microbes over time. We're measuring it based on the DNA present present in the soil. So the Haney test, our results were really taking different lenses, and that's just one area where we overlap with the Haney test. Generally, we don't, but we look at that same sort of concept of microbes that can perform respiration. And in samples taken from compacted fields, we've seen this time and time again. We typically see maybe aerobic respiration is medium to high in the first inch or so, but then it really tends to drop off. In Rick's samples, we saw the exact opposite, where we saw high respiration down low into the soil profile, as shown by the dark blue squares here.
Look at the 6 to 12.
And then that was, so that was a little bit down there. We saw all the way. So we didn't actually include all the data just to make it a little bit more succinct. But I think in the 6 to 12, we had seen similar numbers as far as aerobic respiration, not off the board.
But you see, Gus, okay, folks, to me, this verifies. I mean, I've always said you've got to look for validations that what you're doing is correct. So I can go out with a spade and I can see aggregate stability 10 to 12 inches deep now on this farm. This right here proves it, right, Gus?
Exactly. And just like with the aerobic respiration, that was enough proof in the pudding. Really, we wouldn't, if there wasn't much oxygen getting down there, these microbes wouldn't be able to live at the levels that would be within the high ranges of our database. Then fermentation and methanogenesis, which we would expect to increase with depth in compacted soils, showed really, not generally completely different trends. Fermentation actually went down, which is the opposite we would expect. Methanogenesis went up and down a little bit, was a little bit higher at 3 to 4 inches, but generally showed just, consistent trends. So this really is the proof in the pudding. The DNA doesn't lie, and we know, looking at these levels of microbes and well-established relationships with oxygen and soil permeability. I was glad to hear, Rick, you mentioned when we first visited this data that looking at a tensiometer or a measurement of your soil structure, you're really seeing very solid, spongy texture and not really experiencing any compaction just as far as your physical measurements. So I'm glad to see the biological data really, align with that.
See, to me, this is huge, folks. This is another way. And Gus, I mean, just a real quick answer, because we got questions piling up here, but are we going to be able to do this at the field level and can we do this with a DNA sequencer one day? Can we get to these kind of answers in a few minutes rather than several days?
So there's always, a technological curve when it comes to this. And what I've been really excited about, I've been at a Biomakers almost 2 years now, and there's, when it comes to the sample analysis, it's not as with DNA sequencing, it's not as cut and dry as extracting chemical, nutrients from a soil sample or using an extraction method to measure pH. We have the sequencing method that actually characterizes the biomarkers that we detect in the sample, the microbial DNA that can indicate what species are there. That alone takes about 3 days. The sequencer needs to run for roughly 72 hours, even a little longer. And then on top of that, we also have a quality control process that can generally our turnaround time, Rick, and I don't know actually what it was when these samples were taken, but generally it takes about 2 to 3 weeks currently. And it can take longer in the cases of when we have quality control issues, meaning that when our lab team goes to extract the DNA and sequence it, if they don't get enough fungal or bacterial DNA, they'll actually rerun your sample, and that can that can add on a few days to weeks depending on the process of how much DNA is there. In just my short career, this technology has really sped up and continues to. In the time I've been at Biomakers, our turnaround time has actually gone down by several weeks on average. Our lab team has really done an incredible job optimizing the processes. And there's starting to be some sequencing equipment that is actually smaller and more scalable to field settings. Also equipment to help with the extraction of the DNA, because once that DNA can be extracted and is stable, it's a lot easier to get it, to a lab to process and can take care of some turnaround time issues. So we're moving in that direction, Rick, for sure, of making this more accessible and it's gonna scale up with time. But I'm just just a couple of years ago, as you know, the general science behind it was not as optimized for large scale, high throughput sampling, and it's only getting better with time.
Right. So right now, Gus, is the biome the bee test? Is it fungal and bacterial? Is that the two? There's no protozoa or no nematodes. Is that correct?
No nematodes. That requires a different biomarker than our processes are mainly focused on fungi and bacteria, really the sort of foundation of that food web in the soil. But we can actually detect some protists, certain species that are related, relatives of fungi like Pythium, Phytophthora, as well as Archaea. We can detect pretty easily because they share the biomarker with bacteria. So yeah,
Okay.
mainly bacteria and fungi, but a few other microbes as well.
All right, let me just hold because I got to see where we are on questions here because I'm sure there's questions that are pertaining to what we're talking about. Ed Bourgeois is on. Ed, how you doing? He's on every time. He's one of our great fans. Because of the recent rhizophagy cycle research, can biomakers test the bacteria around the roots and then test in the plant? And most important, what ends up in the seed?
Very good question. I've been really excited by a lot of the research by experts like Dr. James White in the rhizophagy space, Dr. Mary Lucero. There's so much interesting aspect in looking at those bacteria and fungi that can live in the crop, and that's something we're considering. I've been a little bit one thing that's really exciting, and it does take time to the functional database that we look at. There's a lot of research in the back end that goes into ensuring our data is robust, and our data science team, we're not going to rush anything that's looking into anything that's not well proofed out. But there's starting to be more research on the genes that microbes possess, which help them integrate into the rhizophagy cycles. Factors that allow certain bacteria to essentially dissolve into the root once it's absorbed, and similar with fungi. The problem is it takes time. Our data science team builds our database based on peer-reviewed research that's been well established and verified. The rhizophagy cycles really come into focus in the last few years, and as more research comes out on those genes that microbes use that are involved in both the plant and the microbe side to be absorbed into the crop, I'm hoping that there's some aspects we can start to tie in. What's really interesting, we already look at on our tests microbes that are nitrogen, phosphorus, and potassium consumers. And we know now through Dr. White and other researchers, microbes can essentially compile these nutrients, assimilate it into their biomass, and then potentially share it with the crop once it's absorbed. So we already have some metrics that relate to the rhizophagy cycle in terms of microbial assimilation of these nutrients. But I'm excited to see where the route goes as more research comes to light. And we don't offer it as a service. We occasionally do some testing on the microbiome as kind of an ad hoc service, testing seeds or plant tissues for the soil, the microbes present. And I think that's an area where we're seeing increased demand due to interest, not only in the microbiome of the soil, but the phyllosphere, the plant tissue microbiome.
Yeah. Now, Marie, I got a question for you, but just before I get there, I want to go back to Mark's question earlier. Mark, I think this is telling us that tillage is not a good thing. That's how I'm looking at it. I mean, look at the organic matter release. It just increases all the way down through the profile because we're not disrupting any of this soil anymore. I look at this as an extreme check mark of, oh yeah, what we're doing is working. And Marie, how would you, if you were, you had your group of agronomists today, and if you were to show them this, what would you tell them, and what's their reaction?
Do you mean to just like tillage and its impact on compaction in general?
No, just what this chart, this what Gus has just gone through. How would you view this, and how would you talk to this to your group of agronomists?
Well, I mean, in this case, you have a lot of really amazing stuff going on. But usually when we're working with the agronomists, we're identifying the problems. So in this case, I'd be like, hey, you're doing something really great, and maybe you should just keep doing that, and it's working, and it's going deep. And, because obviously we're looking at a lot of blue here, and you've got great carbon cycling, and you've got carbon fixation that goes really deeply, and like there's a lot of good stuff happening.
Yeah.
So when I'm working with agronomists anyways, usually there's a lot of yellow on reports, and we're trying to help them figure out like, how do we
Right.
address low indexes in a way that is going to increase ROI for the grower, improve environmental ecosystem function overall, and like benefit everybody in the system.
See,
So
now what I think you guys ought to do, I've always got ideas here. I think, okay, there's a lot of folks out there that farm in many different ways, okay? You've got some people that just want to do vertical tillage and just work in that top 2 inches. Okay, compare what would their chart here, because they're working the top 2 inches, so I'm assuming the top 0 to 1, 1 to 2, and maybe the 2 to 3 would have a lot more yellow than my boxes do here. And then what about the people who are going deeper and doing, let's say, green manuring and actually, turning over biology into the soil? I mean, I think it'd be great to see, and maybe you guys have already done this, is to compare what happens to these characteristics through the different farming systems, which I think is where Mark was ultimately trying to get to with his question.
yeah, absolutely. We've definitely looked at management practices, like different functionality across management practices, and there's certainly a difference, right? And this all comes from just conversations that we have with growers and agronomists. Is just like you said earlier, it really has a lot to do with context. So understanding the context of that
Yeah.
operation, that region, what's going on in that field in specific. Anecdotally, we can say that there are often trends in a lot of these functions based on management practices.
Yeah.
And we're, working with some larger research groups that are putting together really big studies across the entire United States of looking at management practices as it relates to biological functionality. And there are very strong trends there, right? Like if you have higher nutrient cycling, there's something that you're doing to create that in the ecosystem by working with nature. Um,
Yeah.
we see that all the time. There are definitely strong trends in these functionality indexes as it relates to like soil health practices in general.
Right. If
One
there's any
thing on
more,
Mark's question that I think is key to hit on, Rick, you're not the first person who's pulled samples at multiple depths and had concerns about compaction, and it's usually no-till growers. And we've had where they do have high fermentation and methanogenesis, and the aerobic respiration goes to zero as you go down.
yeah,
So that sounds, and a lot of times it's guys who have been long-term no-till and are considering vertical tillage or something as a tool. So unfortunately, compaction, some folks may not be as fortunate to see such great results and outcomes with some of their no-till cover crop systems, where some degree of loosening that soil may help in the long run. Of course, overdoing it, we also see plenty of samples from growers who do use intensive tillage where things are even more yellow, especially in other functions like nutrient cycling. So Marie really hit an important point that the context is super, super key. Sometimes folks tests don't look, just nice and blue. Other times, looking at depth, there's clear and obvious compaction and soil horizon differences in their samples.
well, yeah, go
Just
ahead.
to
Go ahead,
add
Marie.
like one last thing here too on no-till in specific is that I heard a grower once, this was probably in like 2017, we were out in the field, and I don't remember if this was an original quote from him or somebody else, but he said, "No till, no cover, no good." And so in like, that's a important part of the context. Like it sounds like, and Rick, I haven't heard a ton, I don't, about your operation, but it sounds like you're doing a lot of cover cropping and there's like living roots many months of the year. And that in combination with no till is what makes a system really hum.
Right.
If you like in the plains where we have no till for a real like no till for many years, but there's not living roots and there's a lot of compaction and like there's not you know your the living roots really minimize that compaction and manage erosion, and like, that's how you got down that track. So just
Sure.
just looking at tillage as an isolated practice is not like there has to be more context beyond that to really understand like if it's beneficial, if what else we can do to really get the system working.
Right.
Yeah, that really hits the nail on the head, because I will say, Rick, some of these folks we've talked to have seen evidence of compaction in the results. They're most often not deeply invested in a cover crop program like you, and probably not getting that natural tillage, if you will, biotillage, getting those roots helping establish those channels and form those microbial habitats. So it's the tools in the toolkit and how they fit together. But it makes our lives interesting when it comes to it's actually one of the really enjoyable aspects, is being able to have a conversation and start to tie in some of these practices to the results that we see in the in the biology.
Yeah, I love this. I just this makes me feel good, because it just validates that what we're doing is correct. So I really love this. Ruth Knight. Ruth, how you doing this evening? Thanks for joining us. This question is about the database for microbes. Is it possible for a microbe to express different functionality depending on variable factors? In other ways, or in other words, how predictable or stable is microbe functionality? Also, recognizing that a certain number of microbes may be complex, such as viruses within fungi or bacteria. My audience is extremely well educated. So here you go.
Wow, that's a great question. I know we're kind of up at time, so I don't want to go on too long. But first, one thing I should mention is
there's no time restriction. There's no time restriction.
Thanks, Rick. One thing I should mention is we don't look at viruses. Viruses are very challenging to measure, and they tend to outnumber other microorganisms pretty significantly. So we don't necessarily look at that side. But when it comes to functionality, it's interesting to think about. When we're framing what our results show here, we often explain it as functional potential. There are a lot of factors that influence whether microbes are really going to be performing certain functions. And that's one of the reasons why some of the other data you can and other context specific information is key, because depending on environmental factors, microbes may look great on paper, but may have unfavorable conditions as far as physical soil structure, irrigation, rainfall, temperature. What's good news is that the way that we look at DNA, first of all, for identifying microbes and tying them to functionality, we use the most robust biomarkers that are well agreed upon for identification. And then with the functionality side, what we're doing, that's where the machine learning comes in and is able to match microbes with reference genomes that are established in the scientific literature. And a lot of that research that goes into those to knowing what functions microbes perform does involve validating the essentially how stable those functions are. So if a microbe, maybe a species only, a species has a gene function, but it's really only prevalent in maybe 30% of those microbes, we can see that based on what available full genomic data is available. And then if there's a microbe that possesses, if 98% of the sequence of the full genomes we have in our database show it performs that function, if it's a decomposer or a phosphorus solubilizer, the AI is able to learn and read that and see that it's much more likely this microbe will be performing that function. So I will say it's not perfectly predictable, because microbes are complex. Microbiomes are inherently complex. There's factors like one thing we see, there's horizontal gene transfer, lateral gene transfer going on. Unlike us humans and larger animals, microbes, their DNA can actually be, instead of having to reproduce, their DNA can be transmitted between organisms. So there's a lot going on down there. And that's one thing that's really interesting with biological products, biostimulants. We may not necessarily see that microbe show up in a sample after a few weeks go by, but sometimes we still see legacy effects as far as the microbiome. And one of our hypotheses is that lateral gene transfer plays a role. So I don't want to jump into any tangents, but that's kind of the I guess simpler answer as far as how we match functionality and how predictable it is. And there are other factors as well. Levels of microbial expression may hinge on factors like quorum sensing, microbial ecology. And that's where the functions I mentioned, where we look at microbes that get along and don't get along, that's also able to kind of help hint at that overall community behavior. So yeah, it's a difficult process to make sense of it, but the combination of having database and functional information makes us very confident that those numbers match up with what those microbes are capable of doing in the soil.
Yeah, that's a great question, Ruth. Thank you. Ludmila's on tonight. Ludmila, how you doing? She's been on before. She did a great podcast. Let's now that you've ran a number of these samples for which Regen Ag Lab also ran PLFA tests, you are correct. Can you comment on the correlation between the two tests? Do you know that answer, Gus? Because I do.
I'm curious. I don't think I've seen your PLFA test. So if you have looked at those correlations, I'd be fascinated. I can give a general answer, which is, and we get this question all the time because a lot of folks are familiar with PLFA. Now, we do not have a cut and dry correlation between our metrics. Generally, we both have a fungi to bacteria ratio. That's actually on Rick's reports and anyone who sends in a bee crop test, we have a fungi to bacteria ratio, much like PLFA. But ours, when we're looking at the DNA versus the phospholipid fatty acids. So it's total apples to oranges when it just comes to analytical technique. With PLFA, generally there could be anywhere from 20 to 40% of undifferentiated phospholipid fatty acids. Whereas with the DNA, if we're not able to identify a microbe down to species level, we can oftentimes lump it into a genus if it's taxonomically similar enough. The fungi to bacteria ratio we have tends to skew more bacterially dominant. You may have noticed that, Rick. So we don't often have soils that achieve a perfect 1-to-1 fungi to bacteria ratio. That's simply because the biomarkers that we use to compare, the DNA biomarkers we use to compare bacteria and fungi do tend to be more prevalent in terms of the bacterial population, just based on how many more cells bacteria tend to possess. But the same concept, we do like to see over time as a grower is taking samples to monitor their soil health, we like to see a fungi to bacteria ratio at least shift more towards the fungal side. So same concept in interpretation, it's just apples to oranges, and I wouldn't generalize between those ratios. And one way Lance Gunderson at Regen Ag Labs has described our bee crop test is it's kind of like the PLFA on steroids, meaning the PLFA, which I am a big believer in the PLFA and have used it going back years, but it lumps microbes into what we call functional groups with gram negative, gram positive, looking at different types of mycorrhizae and saprotrophs. With bee crop, we're going much deeper into those functional groups, not just the taxonomy or these gram positive, gram negative groups, but nitrogen releasers, phosphorus solubilizers, carbon fixers, fungicide agents. So we're really leveraging the DNA to look more at agronomic functional groups rather than strictly what the phospholipid fatty acids can tell us. And I believe there are correlations between the two tests. But we haven't necessarily looked at a large enough data set to know if there's any cut and dry patterns. From the small sets of samples I've looked at, all I've noticed is the difference in the bacterial to fungi ratio tends to skew bacterial dominant in the bee crop.
Yeah, and everybody's different. I mean, when I had this notion to do this, I wasn't going to leave anything out. I mean, we did everything. We did it all. So, I mean, this was a very expensive test when you take everything we did and add it together. But look at the data we got from this. And the other thing that I really wanted to find out was, and I don't know, Gus and Marie, I don't know if you guys identified the biology that was at the 0 to 1 and then compared it to the biology that was identified in the 1 to 2 and so on and so on. I wanted to see if not only did we A, have compaction, and B, are we stratifying our nutrients, but are we stratifying our biology at the same time? See, that's where I really wanted to go with this. So did you guys do a biological assay on the 1-inch increment and make those comparisons?
We certainly can. Among the tools in our portal, we have one that allows you to compare species. Now, we often will look at, major plant growth promoters, your functional microbes like Trichoderma bacillus, rather than necessarily looking at the entire population. We also have a tool on our online portal that lets you look at number of species and how many are in common between samples. So that's where I'll sometimes looking at samples that are taken close by, we see a lot of species in common. Whereas if a grower has fields 20 miles apart, there's a lot of difference. So those same sort of tools can be applied here to look at certain specific microbial species and how much that changed. I really liked it, Rick, when it comes to these functions, there's not often just one or two microbes that are driving these nutrient cycles. There's so many there, and there's a lot of complexity going on. So their populations aren't even typically going to be super consistent as time goes on. So looking at these functional groups and how that shakes up from inch to inch really tells me a lot more. Even though I've people sometimes will throw a microbe name at me, and sometimes I know it, I'm at least somewhat familiar with it. A lot of times I'm not, and I have to remind myself no one in the history of mankind can know what all the different millions of microbes do. So that's the benefit of the functional groups. We certainly can, in the tools in our portal, which if we have time could jump on now, we can compare the 0 to 1 inch, 1 to 2 inch, 2 to 3 inch, and see maybe if certain mycorrhizal fungi vary, at depth, and factors like that. So we certainly can do that. It's just the functional groups are often our way of really digging into those agronomic factors that that are going to relate to management practice.
Because, well, I mean, I make some assumptions, but I also am trying to use logic here when I make these assumptions. But I got to think that microbes are just like us humans. I mean, there's certain places we like to go, and there's certain places we don't like to go. And I'm assuming the microbes will do the same thing. So maybe you get time, you can run that test crossways for me and see what's in each of those levels. And folks, just so everyone knows, I'm gonna start putting some of this stuff in my presentations now, because this is the future. And then you take this information, and then you take the groups like what Ludmila's got with her group of biological warriors out there, farming warriors, and now you create these recipes that are prescribed to whatever situation you have going on in your context. And that's where this is all going to be going one day, Gus and Marie, and you guys are doing it there at the lab, but I still think in one day we're going to have something in our hand, and we're just going to be able to slip in the slide, and out's going to come the results, But that's me. So let's see what else we got here. Mark is wanting to give us a little more background on this. Some more background on question: biodynamic versus no-till organic. I'm doing just food grade edible crops. Dry beans, popcorn, sunflowers, and flowers have not been successful with no-till crimped cereal rye on food crops, and have went to biodynamic covers and tillage. Yeah, I get it. Mark. You're way north of us. Your weather's not near as nice as ours. Your season's shorter. I understand all that. And I don't mean to put this. I'm not wanting to shove what we're doing in your face. That's not what I'm about. I'm just excited because all the efforts that we've done are paying off, and this chart excites me that they've got. That they show what our farm's doing. So I know, again, it goes to context, and you're exactly right. So hopefully, Mark, one day we can get some no-till figured out and be just as successful. Michael, I don't have your last name. I'm sorry. I don't know why I don't get everyone's last name. Context is so important. Soil types, soil moisture levels, tillage types, covers, even annual climate factors, etc., create almost infinite variables. Yep, that's exactly right. Folks at Bionutrient Food Association testing for food nutrient density are discovering this also. You bet. I totally get that. And when we start talking about like comparing parts of the country, you can't look at Arizona soil and Iowa soil, and it's not going to be the same. So we have to put these things into context. Gus, what else do you have? You got another slide here for us?
We do. We have a few more slides, and we don't have all the information on your samples here. And this was actually only your wheat samples. I think you also had some sorghum, if I recall correctly. But this next slide, this is looking actually this touches a little bit on your point, Rick, of talking about how not only are nutrients potentially being compacted into layers, but microbes, looking at the population. These three metrics look at the entire microbiome. So rather than sorting them, categorizing them as aerobic, anaerobic, and looking at nitrogen fixers or phosphorus solubilizers, here we have a metric called soil quality, which is our best shot at an overall rating on the biological status. Biodiversity, which is based on the number of species that are present. So biodiversity, we're really just basing it on how many microbes are there, how abundant they are, and how taxonomically diverse or different they are. And then the more uniqueness and the more microbes we have, the higher that rating is. But biodiversity doesn't entirely tell the whole story, because that's telling us who's there and how many there are. It's not telling us a lot about their functions. The resilience indicator on the right, the farthest column to the right, that looks at microbes, that's lumping them in and looking at essentially their ability, the microbiome's ability to deal with stressors and disturbance, to bounce back from tillage or drought or even unfavorable temperature swings. So resilience is looking kind of at the whole community and how redundant functions are and how effectively we'd expect microbes to respond to stressors, both biotic and abiotic. And if there was what you might want to say sequestration or compartmentalization, or however you want to describe it, compaction influencing the microbes and causing the population to defer. What we often will see, biodiversity, once you get below 6 inches and below the rhizosphere, I have noticed just anecdotally, it tends to go down. That's not surprising in a lot of row crops because the roots only reach down to about 6 inches, and below that, it's not dead, but there's a lot less microbes there because they're not getting the root exudates so much. They're oftentimes not as exposed to those disturbances. But your biodiversity, which we tend to see biodiversity correlate with healthy soils in general, as you might expect, it stayed in the high to very high range throughout, even going down to the 5 to 6 inch and even the 6 to 12 inch. The resilience also stayed high, going low into the soil profile. I think, Rick, there's a little bit of deviation here. When you look at resilience, I think we talked about this, but 0 to 1 inch, that's probably where there's going to be some disturbance from, traffic, just from inevitable traffic and harvesting and planting.
A no-till drill runs, the blade is running in that 0 to 1 inch zone. So
yeah, and there's going to be more exposure just to the elements, to air and rain and whatnot. So resilience here is a little lower, but it actually increases from low all the way to very high, going down to the deeper depth. So that's where there might be a little bit of different, but it's really what we'd expect to see, again, because like you said, that top inch is getting some disturbance, whereas down low, this is really what we want to see throughout the soil profile. Is a lot of blue in these overall categories. And then lastly, one other thing that we had touched on, now this is another specific section looking at plant growth promoters or microbes that promote plant tolerance to stressors. So we see a bunch of phytohormones here. We're not measuring phytohormones in the soil. We're measuring the levels of these symbiotic microbes that we know can produce these phytohormones and other metabolites and compounds that help crops deal with various forms of stress. Auxin, cytokinin, gibberellin are kind of your growth hormones, but here we do see a little bit of a higher, more blue in the upper zones, and really here the green boxes kind of highlight it. That's also not necessarily surprising. That's probably going to be where there's a little bit more of those roots. But some of these, do, like heavy metal resistance is a category we look at that measures the microbes that can help mitigate phytotoxicity, mitigate heavy metal buildup in the soil. That stays pretty strong going down. But this was one area where, we do see a little bit higher levels closer to the top, but a fair bit of variability throughout. And that was as far as your samples go, Rick. There's a lot more details to dive into, and I'd be more than happy to take a look at some of those microbial species and groups and see if we do see any differences between the depths. But this slide just kind of touches on all the different metrics. These are all we really break down our reports into 3 main sections, and this encompasses all the different metrics we look at, just to kind of put it all into one slide.
Yeah, this is powerful stuff, folks. And again, if you've got any questions, now's the time. We've got we've got Marie and Gus here. So, and if anybody wants to raise their hand, Rachel can unmute you. Um, let's see, what do you got? What do you got there? HMR is huge. Our food supply is toxic from heavy metals. Yeah, yeah. You know, to Marie, how would you, you know, you and I are sitting down at the table and we're going through these tests. Tell me what your interpretation is of your data here for our farm. Oh, well, just this one field. I don't know if Marie's maybe.
Yeah, I'm still here. Sorry, I've got a couple more hours driving tonight, so I'm back on the road.
No, that's
Um,
okay.
but I mean, yeah, it's just I think Gus really covered it all. We've looked at all the things that are going on, and it's cool when the data lines up with what you see in the field, right? It's just like I forget what guest had the question, but just like how are these things validated and how often do they make sense? And
Yeah.
this is just a really great example of where that happens, right? You have and that's always really exciting, right? And I would say that happens like 80 to 90% in our work, where it's like you see these indexes and then you ask the grower what the context is, and then you're like, oh, that makes sense. That
No.
makes total sense. You've either like today, for example, one of the growers that we were working with had pretty poor nutrient cycling in terms of nitrogen, phosphorus, and potassium, but they've been using like really heavy uncomposted hog manure for like 30 years. And so when you hear that, you're like, oh, that makes sense. Like they had really high or like nutrient consumption because the microbes were just trying to process all the excess nutrients that were there. And their overall nutrient pathways were low because they just had an excess of nutrients, and not the biology hadn't been tended to really process it and like make it available to the plant. So that those are some of the really neat examples where there's something that we can do to move the needle by identifying the root cause issue.
Yeah.
And then we see fields like this where it's like, oh, you're been doing no-till and cover crops and diversity and all this stuff, and then like it's like, oh, that makes sense. So it's
Yeah.
always cool when it then the data really shows us and proves what's going on in the field.
Yeah, not only that, Marie, but what the farmer expectation is. You know, he's assuming that what he's doing is correct. I mean, I'm trying to do my best out here. We're trying to do no-till. We're trying to do cover. And then this is like, oh wow, like wow, yes, thank you, it's working. Uh,
Right.
Claudia is saying, thank you, Marie and Gus. This is going to be an awesome tool for farms, especially regenerative farming. Wow. Yeah. Ed's got a good question here. Rick talks a lot about epigenetics. How can biome makers help with that? That's a great question, Ed. Do you know what he's talking about, Gus? I drive epigenetics home a lot. Do you know what he's talking about?
Oh, sorry, just had to unmute myself. Yeah, I'm familiar well, I'm certainly no expert in epigenetics, but that's something I think a lot about, because when it comes to epigenetics, you talk about how it's really focused on how the environment can influence gene expression.
Yes.
And this gets brought up by some of our clients who are on the microbiology side, with epigenetics, what we're looking at biomarkers that we use to then model and identify microbial species to tie them to a function based on full genomic data in our reference database. But epigenetics is interesting because the genes themselves don't inherently change. That's the expression. Now, I think when it comes to epigenetics, because we're looking at the DNA and it doesn't change typically with epigenetics, that's where we may be able to see dissonance in terms of when we don't, Marie mentioned we do see things line up most, like 80% of the time with what we see on a soil biology report versus what we see in terms of yield and productivity. But when we do see factors where we might profile that, we're seeing strong phosphorus solubilization or strong, strong nitrogen release, strong nitrogen fixation, but maybe not seeing the outcomes or as far as fertilizer use efficiency, or maybe not seeing as much ROIs as far as input costs with biological products. That's where epigenetics is just one of many factors we have to think about. But I think that there's some degree, because we're able to see based on the biomarkers what we expect the microbes to be doing in the soil, that we can maybe kind of take the first stab at seeing how environmental influences, some of that metadata like management practices and region and environment can cause dissonance. But that's a tough question. That's one of the areas certainly on the horizon for us at Biomakers and our data science team is tackling epigenetics.
I'll tell you what I'm going to do there, Ed, and you're more than welcome to give me your ideas on what to do as well. Next spring, when we have actively growing our actively growing maslin, which we've created a cereal rye, a wheat, and a barley combination. I think what we'll do, Ed, is we'll do the shake the soil from the roots of those newly grown plants and have Biomakers run the biology right there at that rhizosphere. So that will be the first way I'll tackle that. If you've got another idea, you let me know. Ruth Knight is back. Ruth's got a question. This is a great question. I'm glad you asked it, Ruth, because I was going to. Let's see, here we go. I am an independent agronomist. How can I access training in interpreting and using the analysis to assist her growers?
Very good question. And I will give a shout out there to a program that we've developed. It's available on our website called the Bee Crop Advisor Program. It's completely free, just a short, about 2-hour course on interpreting using our analysis and understanding how to support growers with it. And it also includes some perks for those who haven't pulled any bee crop tests previously. There's a deal for crop consultants to get a first package of, I think it's 20 or 25 tests at a discount. And it also is a network of crop consultants where we meet twice a month to just go over experiences, have a lot of the conversations about what we're seeing in our results and what we're seeing in the field. So more than I just dropped the it looks like I sent the link, but our Jamie from our marketing team sent it as well. So please take a look at that link in the chat to have some.
Thanks, Jamie. Well, but Gus, do you guys have a do you guys have I mean, school's not the right word here, but do you guys have a class or something that like the Haney test, for example? I mean, that thing's hard to comprehend. You need help going through it. Do you guys do this? Can you do the same thing to teach Ruth so then she can go out and quickly tell her troops, what's happening on these tests?
Yeah, so that's what the Bee Crop Advisor program includes, is that the 2-hour course does focus heavily on interpreting our tests, knowing understanding how to use them. So yeah, we try not to go too deep into the microbiology nuances because we want it to be, accessible and useful for someone who maybe doesn't have a deep background in soil biology, but it's really intended for folks of all backgrounds in soil health and soil biology. And that 2-hour course is a great resource to just get an idea of where to start with interpreting tests. Jamie also mentioned we have an upcoming webinar, and we have quite a few webinars on our website that do go into reviewing data, reviewing results, and understanding some of the interpretation and implications. So definitely recommend those tools on our website. We also have a bee crop guide, which we tend to provide to folks who are looking to get just a reference guide. It's kind of like Rick Haney's regen guide as far as interpreting the Haney test, similar sort of document going through the definitions of the parameters, what's good, what's bad. So we have a suite of resources, but we know that's one of the big challenges with soil health. There's so much complexity in biology and ecology. So we're trying to, it's a big part of our role, and my role at Biomakers really is the educational piece of ensuring that this data gets put into practice. It's a big learning curve, but we're really happy to see so many folks who are wanting to take that leap and really dive in.
Oh yeah, this is big. And you know what we need to do? We'll do another podcast with these folks, and we're just going to have a podcast about going through the sampling procedure. I mean, you gotta collect your samples properly out in the field, and then we'll just Gus, we'll just go all the way. The sample shows up at your lab. What do you do? How do you read the test? I mean, we'll just do a whole podcast on that, and that'll help educate the listeners out there.
That would be great. One thing, I've had one of your reports open in the background, and we have about roughly 36 parameters throughout the whole report. So there's a lot more that we didn't even have time to get into today. But I would be really excited for that, Rick, just to have the chance to go through the sampling procedure. It really is pretty similar to a conventional standard chemical fertility test. But I'd be really excited to go through with your audience, point by point, a report from top to bottom, hit on the areas for potential improvement, what looks good, what looks bad, what's actionable, and kind of just give I'd love to have a back and forth conversation too of just anyone who has any curiosities on what jumps out in a sample report.
Right. Michael, again, I apologize. I can't see your last name. Since the vast majority of crop soils, even organic, are/were impacted by chlorides, pesticides, herbicides that are long-lived, do you see evidence that these residuals might alter these? This is great. The residuals might alter these findings from a normal, non-impacted soil. Is there any consistency with a particular chemical? Not sure. I'm asking. Oh, yeah, you are. You're asking that exactly correctly. That's a great question.
Yeah, I could go on about that one for a while, but I forget, someone else earlier in the chat brought up the complexity and the context, and that's one of the tough factors is that certain soils are just more receptive to holding on to chlorides or holding on to compounds that glyphosate and other herbicides break down into, whereas some soils are pretty good at flushing that out. And then there's also the fact that in region by region, the chemicals, the crop protection products that are going down are very different. So it's hard for us to see any major trends across agricultural soils. But the nice thing is our database, when we're rating these samples according to these functional groups, the rating system is based on our global database. And even for corn, soy, a lot of our samples come out of the Corn Belt in the Midwest, just because that's the big corn, soy growing regions. But because we have such wide variety, including soils from Europe and Australia and Africa for certain crop types, when we see, sometimes we do see things that stand out. And then we get to talking to the client, the grower, or the agronomist, and then it clicks as far as residuals. Some of these impacts of things like chlorides. One of the really exciting ones, and this is also something that's been on my mind a lot, is with glyphosate. And there's a lot of talk about glyphosate and also the product that it breaks down into and the microbes' role in that. And I mean, we've seen enough tests to know glyphosate isn't necessarily fumigating or sterilizing the soil, but there's enough research now to show it has some effects on the microbes, and whether that's good or bad for all microbes, the jury's still kind of out. But we do know that there's been a lot of anecdotal evidence, and also, glyphosate was developed to be an antimicrobial, so it's going to have some negative effects. But if it was sterilizing the soil, like some fumigants are intended to, we know that by now. But one anecdotal thing that I hear come up time and time again, and I think John Kempf, and I think a few of his podcast episodes has hit on this, is manganese deficiency associated with heavy glyphosate application year over year. We just had a grower, I won't shout him out because our data is fully anonymous up until a client decides to publish it or share it, but he had the manganese pathway. That's one of the micronutrients we look at, the microbes that are able to unlock manganese and cycle it. They were super low across a large set of samples. And he said, well, we struggle a lot with manganese, and I see it more of a struggle with growers who've been using glyphosate heavily for a long time, and not so much with those who maybe haven't had to use it quite as much over the years. And I know there's some chemical explanations to that as far as the availability of manganese. And I'm not an expert on soil chemistry or the chemical physical side, but the idea with glyphosate, and we don't need to go too deep into the science of it, but those sort of anecdotal instances, I'm hoping that as time goes by and we get some more data behind it, we're able to start to understand, like we kind of talked about early on, and I think the direction that this soil biology testing is going is with predictive analytics and being able to prescribe and see some of these effects. So as we get more, there's not a lot of folks super excited to test the effects of certain herbicides and pesticides and such. but I'm hopeful that we can start to see some of these factors shake out, that we know we see it anecdotally, like with the manganese and glyphosate, but undoubtedly other factors as well. And I will also just add to that, with fungicides and fumigants, they're really not, oftentimes fungicides are not very selective. We know that they're going to kill some of the good fungi. They may bounce back, but year over year, that may have some detrimental effects. We have actually, I mentioned our quality control process earlier. So if we don't get enough microbial DNA, we have to rerun the sample until we do get enough for it to be valid and meet our standards. We've had quite a few samples, especially from golf courses, which get fumigation or fungicides applied on a weekly or biweekly basis. We've had a lot of samples that were taken right after fumigation or right after fungicide fail, especially in the fungal side of not obtaining enough DNA. So there's a lot to unpack there, Michael, with that question, and we certainly do see impacts. And I think that there's a lot of evidence that the microbes do play important roles in remediating those long-term impacts, but still a lot to tease out in the data as far as cut and dry negative impacts and long-term effects of some of those residuals.
Yeah, that's great. Great question. Great answer. Um, uh, any season to maybe we can get Marie to I know you're driving, Marie, and I appreciate that, and that's what I like about this being live. I want this to be fluid and what's going on in your life. Paul's on almost every week. How you doing, guys? Any season? Is there any season to pull the sample? I get one season, spring, summer, fall, and what's the cost to run some of these tests?
Yeah, so we get this question a lot. The best time to pull, I always say, is like operationally contextual. So, what are you using the test for? What decisions do you want to make using the test? Knowing that it takes a couple weeks to get the results and you want time to make a decision with it. Most growers are pulling the sample immediately post-harvest or early in the springtime. And that's when you're out pulling your fertility sample, and you can do it at the same time. The sampling protocol is near this, like it's the same. It's just that you need to send it to the lab sooner because you're working with living DNA. But the protocol is the same. Composite sampling, top 2 to 6 inches with a soil probe. So yeah, I would say that the best time to take samples is when it makes the most sense and what, depending on what you want to use the results for. And
Right.
and then second piece being to do it at the same time every year. So if you start pulling samples in the fall, then just do it every fall. So you're comparing it to the same general time of the year post-harvest, or in the springtime. And then the cost generally breaks down to around $2 an acre, which I think is pretty similar to grid sampling in crop production. So it's $200 a test roughly, and you're doing less samples. So we sample on a grid or on a zone basis rather than a grid basis, like you might for fertility analysis. So yeah, that's the cost breaks down to roughly $2 to $3 an acre.
So, Marie, would you also recommend that you drop a pin and geospatially mark where you're pulling this sample from every year then?
Yeah, I mean, you're pulling a composite sample, so you're, pulling cores from across an entire management zone, so it could be a 100-acre field. So, the georeference is just sure.
Yeah, okay, very good. And that, what I'm glad you brought that up. And I know I said we don't have a time limit, but we do have to be respectful of time. We're getting close to 2 hours, or no, what are we, about an hour and 40 minutes. What, I like to do, I like to do these tests in conjunction with the Haney test. So,
Mm-hmm.
uh, is that kind of a typical way to do this?
Yeah, absolutely. So that's one of our standard offerings. We call it a B Crop Plus test.
Okay.
We're partnered with Region Ag Labs, who runs the Haney test, and now they actually are running B Crop tests as well in their lab. So we love Lance and all those folks over at Regen. The B Crop Plus test, when you're looking at the biology plus the chemistry, is my favorite way to look at things, because you've got
Yeah.
your potentially available nutrients based on a water extraction, which mimics the soil biology. So that's your chemistry side of things. And then you've got the biology that's actually there to leverage it and to make those things actually available. Um, I think that's the future of soil testing, is looking at both all the time
Yeah.
and using that to inform our decisions. And I'll just add, I don't know, it's just coming up for me that like we work with growers on every side of the spectrum, and this technology and this way of looking at things is highly relevant for regenerative growers, for conventional growers, for everyone in between. And understanding the soil biology and understanding how to make decisions based on that moving forward is the future of agriculture.
Yeah, you're exactly right. And it's nut farmers, it's tree farmers, it's vegetable growers, it's everybody. It's
Yep,
everybody.
100%.
Yeah. Any last questions, folks? Because I'm headed to the finish line here. Marie, you first, ladies first. Take us home here. What didn't we touch on? You know, give us your final thoughts. And we're going to have you guys back sometime soon. But take us home here.
Yeah, I mean, I think I kind of just hit that nail right. And what I always really try to get across is that the looking at the chemistry and the biology together is the next step change in agriculture.
Yeah.
It's coming back to the what I've always said is like it's using age old wisdom about the natural systems and how they work and how they grow giant trees in the forest, and new age technology. So age old wisdom and new age technology to come together to create the next step change in food production and how we do things. And so that's what I get really excited about, is like this intersection of technology and with like, you know, nature and science, and seeing all those things come together. And like that's how we're going to feed the world for generations to come. And
Yeah.
and we've got to, you know, we're at a tipping point, I think, of like it having to really shift that direction. And that's why, you know, this has to meet everybody where they're at.
Yeah.
And that's what we're doing at Biomaker. So it's really exciting.
Yeah, that's awesome. Well, I'm going to get to you in a second, Gus, but yeah, thank you, Marie. That's a great way to wrap it up. And Michael's telling me his last name is Mike Atzit. Thank you, Michael. I appreciate that. Hey Gus, is the side by side with Malik 3 useful compared to the Haney? What's your opinion on that?
It certainly can be. You know, we have folks who have their personal preferences as far as chemical fertility tests, but both Regen Ag Lab and I believe both of our other two partnership labs, Waters and Waypoint Analytical, both offer the Malik. And similar, we like the Haney because it does have that water extractable organic nitrogen, and also looks at phosphorus and carbon through a little bit of a deeper lens. But you can certainly use your standard Malik 3 as in conjunction and side by side, as Marie was describing. Look, you know, on the bee crop test, you have what microbes are present to mobilize those nutrients, and then on the Malik, you're looking more so at what's available or what's there for the crop to work with. So yeah, they can certainly be used in tandem as well.
Yeah, that's great. Well, Gus, oh my gosh, you've gone through a lot of stuff here tonight. Take us, what we forget, wrap this up for us, and we're gonna head for home here.
Well, one thing I want to mention, Rick, is, and this gets back to kind of that context specific challenge in regenerative ag, and also what Marie was mentioning of meeting people where they're at. And it's such a challenge because of the fact that there are no silver bullets. One of the reasons that, you know, a lot of that conventional ag, you know, took the shape it took is because it's very easy to go out and just try to, you know, kill and control and, you know, take a problem and match it with a solution and just kind of think inside the box that way. But with regenerative ag, suddenly the box is open and you're thinking from a systems approach. Everything's interconnected, and you have to, when you try one thing, you have to think about, you know, not just the direct thing you're trying to find a solution for, but also the indirect effects and the other implications. So it's a challenge. And I think, Rick, folks like you who are on the pioneering side of regenerative ag, who have platforms like this to share your knowledge and experience, to me, that's hand in hand with the data and the information that folks like Biomaker's Regen Ag Lab provide. Folks like you who are out there and experimenting and pioneering regenerative practices that work, and especially on large scale row crops. To me, that's it's super important just to ensuring that folks do have the tools in the toolbox. You have to start somewhere, but it's daunting when there's no one in your region, or you're trying to, it's risky as growers, and there's kind of a vicious cycle to a lot of conventional agronomy when it comes to even things like crop insurance, but also just inputs and kind of staying on that route.
Yeah.
Turning that route around, it takes tremendous confidence, a tremendous effort to learn. And I just wanted to end by hitting on the tremendous role that you play, other folks that we've talked about, like John Kempf and Steve Groff, other pioneers in the regenerative space. It's tremendous to have resources like you. And I know we've touched on a lot of topics today, but it's been great conversations and great questions from your audience. So yeah, appreciate the opportunity, and thank you for all the work you do getting the knowledge and expertise out there.
Well, thank you. I appreciate the kind words, and I have the most educated audience in all of podcasts. I have a great group of folks that travel with us every week, and they keep me on my toes, I'll tell you that. So thank you to everyone who came on tonight. We're getting all kinds of thanks. Happy Thanksgiving. This has been a great session, a lot of information, super information. So guys, thank you. Marie, I know you're busy, you're headed for home. Please be careful getting yourself home. Gus, thanks for coming on with us tonight. This has been great. I've been wanting to do this for a while with you guys, because you guys are like a sleeper out there, then all of a sudden you're just going to turn into a giant. So this is great stuff. Keep up the good work, guys, and thank you much. Appreciate it.
Thanks for having us on, Rick.
Yep, you guys are welcome.
Appreciate it. Have a great night.
You're welcome, and happy Thanksgiving, everybody. And I got to. What is next week is Thanksgiving? Is that right? Oh my gosh, it is. So there will be no podcast next week. We're going to take a week off. So everyone, have a great Thanksgiving, and we'll see you soon. Thanks, guys. Bye bye.